Getting to know the biological diversity of our planet is an important task, especially in the times we live in, where the rate at which species go extinct is higher than the rate at which we discover and study them. This is no different for parasites, and they could be in an even worse scenario, because virtually every free living organism on Earth has parasites. Mammals, lizards, plants, starfish, you name it. So, if a species goes extinct, it is not only that species the one we are losing, but its parasites as well. You may be grossed out by them and wonder why we just don't eradicate them all. But parasites are also living beings and represent important nodes in maintaining rich and resilient biological networks, controlling the population of their host or serving as food for other organisms. In addition, parasitism is one of the most successful and diverse ways of life, estimated to account for around half of all biodiversity on Earth. Today, we're going to have a really close interview with bats again, but instead of asking them if they have a close relationship with dengue virus, we will ask them which parasitic nematodes they harbor. Plus, we're going to see how this contributed, albeit just a tiny bit, to the establishment of a natural protected area. So, this is a pilot study we carried out way back in 2007, where we were cataloging the diversity of parasitic helminths of a colony of bats in the Salitre Cave in Mexico. We were looking for all kinds of worms, tapeworms, flukes, nematodes. I was in charge of looking specifically for parasitic nematodes of the digestive tract, and other colleagues looked for tapeworms, also in the digestive tract, or cysts and larvae of these parasites in the rest of the bat's organs. Our ultimate goal was to determine the ecological importance of the Salitre Cave, which we already knew was quite important because it is a maternity cave, where thousands of bats give birth and nurse all at the same time. This alone makes it a central hotspot for conservation biology and the overall ecology of the zone, and has to be preserved and taken care of just because of this. But a proper biodiversity study of the region will add much valuable information to support the rise of this cave and its surroundings through a natural protected area status recognized by the state. In addition, the knowledge of the kinds of worms that parasitize the bats living there allows for an estimation of the ecosystem's trophic complexity and perturbation degree. This is because parasites with complex life cycles, like those that require multiple host species, may be, depending on the context, more vulnerable than other parasites that only have one or two hosts. So, heavily perturbed ecosystems may present simpler parasite species. Given this, my task was to create a registry of the parasitic nematodes found in bats inhabiting the El Salitre cave, and to determine the prevalence, abundance, and intensity of their infestations. That is, the percentage of infected hosts, the amount of parasites per host, and the degree at which the hosts are infested. But what are nematodes? Nematodes, or roundworms, are vermiform and unsegmented organisms ranging in size from a fraction of a millimeter to several centimeters or even meters. They are free living organisms known to live everywhere, from the summits of the highest mountains to several kilometers underground in caves and mines, and from the coasts to the abyssal regions of the sea. But, of course, there are also parasitic species in virtually every group of animals and plants. They are present in all these places not just as a handful, but actually are the predominant type of animal both in terms of numbers of organisms and species, representing about 80% of all animals on Earth, with more than 1 million estimated species. It is worth mentioning that this study was only part of a larger project, in which plant biologists, ichthyologists, herpetologists, entomologists, ornithologists, anthropologists, and sociologists, each within their own areas of expertise, of course, provided evidence of the importance of safeguarding the ecological processes that take place in an area of 1,500 hectares around the cave and that have a direct impact on it. So, the El Salitre Cave is located in the municipality of Tlaltizapan, in the state of Morelos. It forms part of the Sierra Madre del Sur and probably was originated from subterranean lava flows. However, the cave has a lot of karstic activity, which means that there is dissolution of the limestone bedrock as a result of water filtration, making it an ideal habitat for bats due to its high relative humidity. The area is surrounded by patches of deciduous forests, farmlands, and paddocks. Unfortunately, there's a lot of human activity in the surroundings, with farmlands slowly being replaced by these big monolithic concrete foundation slabs, where people build massive residential complexes that are completely out of the ecological context, endangering the bat's habitat. Fortunately, the owner of the land on which the cave is located, Jose Varela, is keen to preserve the cave and has gone to great lengths with the help of the Autonomous University of the State of Morelos to protect his farmland from land investors. The cave has three chambers, one located to the left of the entrance, and the other two at the end of a long tunnel to the right of the entrance. A bunch of animals live in the cave, but as far as bats are concerned, up to 11 different species have been found roosting there. The easiest way to catch them is to place the mist nets right at the entrance of the cave. So when the bats try to leave out at sunset, they will be caught in the net instead. So we did that, and to our surprise, not many bats fell into the nets. They were avoiding the net and didn't want to come out of the cave. A couple of hours later, we ended up catching about 17 bats of 4 different species. This was in May, a month after the arrival of the pregnant females to give birth. Of course, we released the pregnant and lactating ones, and placed in cloth bags the remaining 13 bats for their herpetological examination at the Wild Animal Parasitology Lab of the Autonomous University of the State of Morelos. The bats were kept alive until the next morning, when we carried out the herpetological examinations, and yes, for this, the bats had to be sacrificed and dissected. By the way, we found that one of the bats escaped our palpation examination, and we ended up finding this fetus, which I still preserve to this day. Okay, let's get it back to its resting place. I don't want it to get damaged. Since I was the only interested in nematodes of the digestive tract, I first examined the mouth and perineal region for the presence of parasites, and then removed the rest of the digestive tract, from the esophagus to the rectum. The rest of the body was given to my colleagues to look for parasites in other areas. So, to find nematodes, what you have to do is to cut open longitudinally the entirety of the digestive tube by tearing it with dissecting needles or with RE scissors. For this, I placed the digestive tube in a petri dish with a little bit of saline, just to keep everything moist and parasites alive, and then I just started cutting, like cutting a straw to make a ribbon. Using the dissecting needles and a very thin brush, I then transferred all the nematodes to smaller petri dishes as I was finding them. Now, to study them, the nematodes have to be fixed in an almost boiling hot 4% formaldehyde saline solution. This fixative has to be really hot to kill them instantly and prevent them from curling up, which is important because curled nematodes look like nuts under the microscope, making them more difficult to study and identify. Then, using a heating plate at 40 degrees Celsius, such as this one, the nematodes were dehydrated and clarified by placing them on microscope slides and gradually increasing the concentration of glycerin solution. Once clarified, cover slips were placed on top and the nematodes were measured with an ocular micrometer and identified with taxonomic keys. I also made anatomical drawings to scale as accurate as possible using a microscope drawing tube attachment. This device is nothing more than a camera lucida, which allows you to trace on a piece of paper what you are seeing in the microscope. I know we could have just taken pictures to measure and trace the parasite in the computer, but the professor encouraged me to actually look at the parasites for a long time, like really looking at them, to measure them directly and to draw them, first with pencil and then with stylographic pens with different nib widths. It was a hard work, but to be fair, worthwhile. Anyway, so what were the results? In total, we found 320 nematodes from 5 different species, but almost all of them were from this one single species found only in Myotis vellifer. And Leptonicteris yerbabuena didn't have any parasitic nematodes in its digestive tract at all. This is interesting because this bat feeds on the nectar of flowers, and I may be wrong, but as far as I know, there aren't that many parasitic nematodes in flowers. Glossophaga oricina, where we found only one nematode in one captured bat, also feeds on nectar, but has a less restrictive diet and also eats fruits and insects. Myotis vellifer and Pteronotus parnelli, on the other hand, are insectivorous and show a higher number and diversity of nematodes. Nevertheless, our sample size was really small, and our results do not mean that nectar-eating bats do not get infected. Actually, we found flatworms in all of these species, and other studies have found nematodes as well. It just makes me wonder if highly diet-specific bats tend to have fewer parasites, and if they do get them, if these are the kinds that only require one host to complete their life cycle. Most likely, these bats are getting infected from water, from the wet rocks of the cave, or maybe when they groom each other as if by a fecal oral route. Another interesting point is that none of the bat species shared any nematode species, suggesting either that these parasites are host specific, which they are to some extent, or that the bats eat different kinds of insects, take water from different bodies of water, or because of their segregation inside the cave while roosting. So the first parasite, Alintosius tadaride, was found in large numbers in the gut of Myotis velifer. At first, we put these nematodes into the genus Tadaridanema, or Tadaridas nematode, but upon further inspection, I realized that they didn't belong to this genus, mainly because the posterior end of both males and females is quite different from those of Tadaridanema, in which the females have a caudal spine with caudal lobes, and the copulatory bursa and spicules of the males are more elongated and less rounded. The copulatory bursa is characteristic of strongylid nematodes, and is a structure that allows the male to grasp the female during copulation. Once the female is firmly grasped, the spicules are then inserted into the female's vulva to make room for the sperm to enter. Anyway, it was clear that these parasites were trichostrongyllids because of the cephalic vesicle, but I wasn't sure if they would fit into the Molinae subfamily, where Tadorida anema is classified, because of the lack of the spine and caudal lobes. In fact, this will place them in a completely different subfamily, either Trichostrongylinae or Elegmosominae. I looked first among the Trichostrongyllidoids because they certainly lack caudal spines and some had cephalic vesicles. The taxonomic keys I used, which I find really dense and difficult to use, led me to nematodes like hemomonchinids, like this Ostertachia. However, these nematodes lack a cephalic vesicle, and females usually have a vulval flap. The spicules are also very different. Or to the Cooperinids, like this Impalaya, which have a cephalic vesicle differentiated into acephalic and cervical portions, like in the one we are trying to identify. However, these nematodes are mostly parasites of ruminants and other animals in Africa, so it is also important to consider the type of host being parasitized. Then, at some point, I was directed to another subfamily, the Legmosominids. Here we can find Travasostronghylus, which, as you can see, is pretty similar. Well, maybe not so much in terms of the copulatory bursa and spicules, not to mention that the females have a vulval flap again. But we were on the right track because within the Elegmosominids, there is this group of nematodes called the Ornithostrongylinids, which the professor I was working with suggested that the parasites we were working with belonged to. Ornithostrongylinids are, as their name suggests, mainly parasites of birds, but there are also species that parasitize exclusively American rodents and bats. Specifically, he said they belonged to the species Alinthosius tadaride, described by Caballero in 1947 in Tadarida bats. And he may be right, I mean, the description fits almost perfectly, and the females lack caudal spines or lobes. But there is one caveat. The females of the genus Alinthosius, as first described by Kidwood in 1937, have a caudal spine, so I don't really know. I'm afraid that what I should have done was to make cross sections to see the arrangement of the thin lobe, and to unfold the copulatory bursa to take a closer look at the bursal rays, which are of taxonomic importance. Maybe someday I will dive into the mythological collection to find these specimens and revisit them. Anyway, here's where you can find it in the Tree of Life. And now that I'm seeing this tree again, it kinda reminds me of the Gustav Klimt's Tree of Life. He must have been into something. Anyway, I won't show you this tree again for each parasite because all of them are inside this little branch. And finally, this is the description, so if you're interested, pause the video to read it. I know this video is getting pretty technical, but the next parasites will be easier, I promise. The next species of parasites found in the stomach of Myotis velifer was a trichocephalid, so called because their anterior end is super thin like a hair or a capillary, hence their other name, capillarids. Oh, beautiful trichocephalids. Their classification is a total mess, even more than other nematode groups. I just hate it. People can't decide whether their family has only one genus, Capillaria, with more than 753 species, or the same number of species distributed in 20 genera. These parasites like to eat flesh rather than the contents of the digestive tract, so they live inside the lining, feeding on it. Perhaps that's why they have this super thin anterior end, so they can bury their little heads in the tissue to feed on it. Like strongylids, these nematodes also have a copulatory bursa, although much smaller and simpler. and only one, often very long, spicule protected by a shed, or even the lack thereof altogether. They have the esophagus divided into two parts, a muscular and a glandular esophagus. The glandular esophagus has these specialized glandular cells called stichocytes, which form the stichosome. They also present a structure called bacillary band, which is a modification of the epidermis, and it is thought to aid in tissue digestion, although osmoregulatory and nutrient acquisition functions have also been proposed. I lean towards an adhesive/attachment function. To me, it seems quite plausible, given that it's derived from the hypodermis, and has a glandular portion with cuticular pores that might as well secrete some kind of adhesive. In any case, I classify this one in particular as Capillaria palmate. Its description is almost totally similar to that of Chandler's. Nevertheless, I like to follow the classification of Moravec, and later Hoda, and place it in the genus Oncotheca. This is because, according to Moravec, all capillaries that have caudal lateral ale and a membranous bursa with lateral lobes, in addition to a smooth or lobed spicular shed, as opposed to a spiny shed, should be classified as Oncotheca, so that's what I did. Here's the full description. Okay, so now the parasitic nematodes of Pteronotus parnelli. As I told you earlier, this bat is also insectivorous, and we found two species of nematodes in it. The first one was also quite difficult to identify, and that's because the taxonomic keys took me all over the nematode taxonomy. This parasite has such generic traits that it could be placed anywhere at first glance. The fact that we only found females, or females in two bats, didn't help that much, because females often look the same between species, and are the males the ones taxonomically important. However, there are nematodes where the males are unknown. In other cases, the males live inside the female's uterus, yes, inside, like parasites, fertilizing the eggs directly. This was not the case though, and none of the females presented eggs, so I think these nematodes are not parthenogenic either. That is, when the females do not require males to reproduce, producing clones of themselves instead. Furthermore, they are also not hermaphroditic, as no male organs such as testes could be seen. These nematodes were fairly long, with an average length of 55 mm and a width of 0.42 mm, which means that these nematodes are as long as the bats, which measure about 70 mm long. The anterior end is rounded with a buccal capsule and a cylindrical esophagus. They are monodelphic, meaning there is only one uterus, with the vulva located about 2/3 of the way down the esophagus. The anus is at 0.58 mm from the conical and rounded tail tip. All specimens were found in the mesentery, this folded membrane that attaches the gut to the abdominal wall. Based on this information, it is difficult to make a definitive identification of the nematode. However, we can rule out the Rhabditina suborder, which typically has a different type of esophagus. Instead, the cylindrical esophagus suggests that this nematode may belong to the Spirurina or Tilenchia suborders. That being said, there is a possibility that this nematode belongs to the Dioctophimatidae family, related to the trichocephalids, which includes nematodes with cylindrical esophagi and monodelphic reproductive systems, with the vulva near the middle of the esophagus. Not to mention the length, which can reach almost a meter long. Besides, the nematodes of this family can be found freely in the coelomic cavities. However, as far as I know, there haven't been described Dioctophimatids for bats. That's when I went back, and turns out that within the order Spirurida, there is a group of parasites that are characterized by living in the body cavities, the oncocercinids, and within these, there are some that are quite long, up to 250 mm, monodelphic, with cylindrical esophagus, and a simple rounded anterior end with a buccal capsule. In the end, I narrowed it down to the genus Lithosomoides, because these infect American bats, unlike other genera of the family Oncocercinidae, which infect African and Asian bats. Lithosomoides is closely related to Filaria, and it is therefore transmitted by arthropod vectors, in this case mites. So here we have our first vector-borne parasitic nematode. Nice. It's really strange that we didn't find a male though, since these nematodes have males for sure, so it must have been bad luck. Again, here is the description. The next parasite of Pteronotus parnelli, Websternema parnelli or Webster's nematode of parnelli bats, was fairly easy to classify because of its cephalic armature, which is quite common in bat nematodes. Like Alintosius, it belongs to the family Triconstrongylidae. But for some reason, Hoda in 2022, who undertook the enormous task of writing the latest index of valid genera under classification, placed it in the subfamily Triconstrongyline, whereas I will regard it as belonging to the Molinaeinae subfamily, because, among other things, The female has caudate lobes and a spine. Also within this subfamily, we can find all those parasitic nematodes of bats that have the characteristic cephalic armature, as in Biacantha or Istiothronchilus. In fact, Websternema was previously classified as Istiothronchilus. Nevertheless, nematode taxonomy is a mess, it's just a mess. Even when I was studying this at university, the classification was constantly changing. The only difference between the nematode we found in this study and those described by Webster are the striations in the cervical region and posterior end of the males. However, this could also be an artifact of fixation. I'm leaving the full description here if you'd like to read it. And finally, the last nematode, found in Glossophaga oricina, I just don't know what it is. It belongs to the family Capillaridae, that's for sure. It was found in the stomach, like the previous Capillarid. It has a stechosome, one bacillary band, maybe more, telomocytes at the end of the esophagus, the vulva at the end of the esophagus with a vulvar appendage in the anterior vulval lip, conical tail with a spherical end, and eggs without polar plugs? That's a new word for me. Anyway, I only found one female and no males, so I guess it is almost impossible to determine the species without molecular data. Because as I said before, in capillarids, the posterior end of the males is the main feature to consider for classification. So those were the results. The idea was also to calculate the nematological biodiversity of the community using Shannon, Simpsons, and other indices. But with the sample size I have, these are all meaningless. There is just not enough data. To make these values representative, I would have had to collect more bats, but regrettably, I abandoned the project and moved on to studying malaria parasites instead. That's why this video doesn't have a proper conclusion. There is nothing to conclude, except that these parasites can be found in this species of bats. Even then, it is possible that this parasite-host species combination are accidental. Nevertheless, a couple of colleagues continued with this project. One of them calculated these indexes for the helminths of Myotis bellifer, and the other one, my friend Jorge Peralta, ended up describing I think 5 new species of nematodes, and one of them, Spiroera mexicana, found also in Pteronotus parnelli bats, was discovered in the Salitre Cave. As far as the Salitre Cave is concerned, last week was the 5th anniversary of its designation as a natural protected area, celebrated with activities for children and the presentation of a video about the bats of the cave. I was very happy to find out about this designation while making this video, and that studies like those by Peralta and Said Ramirez, who finished what we started, contributed to this. However, instead of the 1500 hectares intended to be protected, only the entrance to the cave was declared a natural protected area. Not even the surface area covered by the cave, just the entrance. This means that if someone wants to build above the cave, they can. It's almost a joke, but hey, it's something, I guess. Before I sign off, I want to share my strong desire to turn this channel into a platform for showcasing my research and sharing my discoveries and insights with the world. My goal is to create content that goes beyond educational explanations and incorporates my own experiments. That's why I am excited to announce that I am in the process of setting up a home lab that will enable me to continue pursuing my passion for nature and explore exciting projects that I have been eager to share with you through these videos. But let me tell you that I have a lot of projects waiting to be realized. However, I recently received an ultimatum from my university, which means that I will need to prioritize completing my doctorate degree. While these may mean a temporary hiatus from posting videos, I mean they haven't been regular or anything, I remain committed to this channel and look forward to continue sharing my work with you as soon as possible. In the meantime, if you enjoyed this video and would like to support my work, please consider sharing it and leaving a comment. Your support means everything to me and will help me get this project going. Thank you for watching. Bye.